move on to the next. So Louis Pasteur. So who was it? Louis Pasteur. He was the father of microbiology. Who was he? The father of microbiology. Okay. So what did he do? He discovered the two things. One was pasteurization, one was your fermentation. Okay. So firstly, what is this pasteurization? Pasteurization is nothing but for whatever. Do you drink a raw milk? No. Why? Because there are some microorganisms and some unwanted substances, little organisms present in that. So you heat it to a certain temperature. So once you heat it to a certain temperature, all the unwanted toxic substances will die in that. So that is called as pasteurization. Heating of a substances to kill all the microorganisms. That is known as your pasteurization. So you heat the water also sometimes. Okay, you heat the water and drink when you are not well. Why do you drink a hot water? Because you feel that if the water, what all the contaminated organisms, minor microorganisms, it may not do you, very big amount of issues, but still if you heat any substances to its boiling temperature level, he said that all the microorganisms and any other toxic substances will die. That is known as your pasteurization. Heating of a substances to a higher degree, that will die to all your toxic substances. So next is your fermentation. So what is this fermentation means? Where it is a biochemical process. Okay, what is fermentation? It is a biochemical process where your chemical substances, it is a breakdown of your chemical substances. Okay, that will break your chemical substance. For example, you will have a sugar. So that sugar is break down into a alcohol. So what is this? That is biochemical. Why? Because it is in biology the chemical reaction is taking place. Biochemical substances. For example, you have a sugar. That sugar is break down into a alcohol. So where it is happening? In your wine. Okay. For example, you do it in your grape juice. Once the grape juice is done and you keep it for a long time, some 1 week or 2 months, maybe you would have heard like it is like 6 months, 1 year, it has a good high content of alcohol and it tastes very nice. Okay. So that is what? Wine. In this wine, you are using grapes. You leave it for a longer time, what happens? It gets fermented. It is an anaerobic process, is the breakdown of your glucose, that is nothing but your sugar. So what happens in fermentation? It is a biochemical process, the breakdown of chemical substances into that will give you a conversion of your alcohol. In wine, the sugar is break down and it is giving rise to the alcohol. That is your fermentation of fruit by yeast through producing alcohol in wine. Okay. So now you got to know what is pasteurization and your fermentation. Okay. What happens in some preparations, they will add the yeast also, as I said for bread, and there are so many yeast that will give you a fermentation process. If you want to do something fermentation, your yeast has to be added. Okay. So now let's see. So what happened in the 18th century? It was observed that the fermentation could be of 3 groups: evolution of gas, formation of alcohol and product of acid. When the fermentation has performed, they used to get lot of gases, that gas and your alcohol and acid. So that was they observed it in the 18th century, and later on they wanted a glycerol for the World War. Okay, they wanted to preparation of your atom bombs. So they wanted in a huge quantity. So what happened? So see, when there are preparation of a huge amount, so they invented this bioreactor. In this bioreactor, they started preparation of your glycerol gas for the atom bombs. Okay, so and also in this bioreactor, your penicillin. What is this? Your penicillin is your antibody, antibiotic. It is your antibiotic. So when you get fever, you usually take, right, antibiotic. So that was also founded in by your founded in the sense it was done, from your bioreactor. So from this they did your glycerol glasses in a huge amount for your world war. They need to do atom bombs, and also the penicillin was production was done in this bioreactor. So next, let's see. So who was a modern father? The term biotechnology was given by the other scientist. So here father of biotechnology was Zacharias Stansin. He was a father of biotechnology. He founded the microscope. Okay, microscope where you can see a minute organisms which you can't see through your naked eye. So next, actually there were so many scientists who had come up, they'll come up with their ideas, and before this also there will be so many one who is working on that, but with the final results, the one scientist who had come up. So I'm just giving you that overall view. Okay, so next one is a Robert Hooke, a physic, examined a cork. So what happened? You have a cork, in that he took a thin section and he saw that in a microscope, and he named it as a cellulae. What did he do? He named it a cellulae, called as cellular. He named it as a cellulae. The next one was your Antony van Leeuwenhoek, a Dutch shopkeeper, saw living organisms. Okay, he saw in pond. You can see some green things, that is not dirt, that is also a living organism, your algae. So in that he saw it in a microscope, and he called as animalculus. What did he named as? Animalculus. Your modern father of biotechnology and the term biotechnology, and your microbiology father Louis Pasteur. He founded pasteurization and fermentation. Okay, so he told us, he named it as animalculae. Okay, so let's move to the next. Rudolf Virchow, a German scientist, he concluded that a living cell is always existed from the pre-existing cell. So for example, you have this cell, now this cell is there. So how did this cell arise? From its pre-existing cell, pre-existing generation, the cell which divides and gives us to two cells, like that only. All the cells exist from the pre-existing cell. So that you can, the term tell also as your cell theory. Okay, existed from your pre-existing in 18th. Pasteur developed the process of pasteurization. I already told you what is pasteurization, boiling of a substance to its particular temperature, that is your pasteurization. The next coming to your scientist is Gregor Mendel, who was the father of genetics. Okay, this is very important in your biotechnology. Who is the father of genetics and your father of microbiology? The one who is the modern father of biotechnology. You guys have to just remember that, they may ask you in the questions also. So the Gregor Mendel was the father of genetics. So Gregor Mendel, he was the scientist who studied both your biology and mathematics. Okay, he was the one who was first person to apply your maths in biology. Okay, so he applied your maths in biology, and he almost worked for 7 years for this experiment. So what did he do? Let's see. The first person to track one trait through several generations. So what is this trait? Is basically nothing but your characteristics. Trait is nothing but your characteristics. So he did an experiment on the pea plant. Okay, he also grew and studied pea plant. So he did what he did from one trait, one characteristics, he gone through a several generation. So he took a pea plant and did so many experiment on that. Well, you may think, why only he took a pea plant? Just because it was available and he won't? No, not like that. Because the pea plant has several contrasting characters, like it was tall and curly, wrinkle, and so many contrasting characters, and the flowers also. So he selected a pea plant, and since it has a shorter lifespan also, so he did it and in one characters he just went through several generations. So let's come to our next scientist. So Alfred and Hershey and Martha Chan. He was the one who founded out the DNA. He told the DNA as a genetic material. He was the one who told the DNA is a genetic material. And the next come your J. Watson and Francis Crick proposed double helical structure of DNA. They was the one. DNA is double stranded. Okay. So they were the one who proposed the structure, but he was the one who found out the DNA is a genetic material. Okay. So now let's see your overview of your biotechnology. So what happens? In biotechnology, you have blue biotechnology, green, red and your white biotechnology. So as usual, your blue will tell you your aqua and marine. Okay. So your green biotechnology is nothing but your agriculture, and red is your medicine. White biotechnology is your industry. So what does this marine, how does this biotechnology, it is taking place and for performing in your marine, in the sense that will give a help in the improvement of certain, nutritious to the fishes, and also it will regulate some of the poisonous fishes. Okay, next coming to agriculture. Obviously what in agriculture? Biotechnology or pesticides, and so many you will be studying from BT plants, biotechnological plants. You are resisting the insecticides which is affecting your plants. Okay, and in your medical, your vaccines, okay, and your antibiotics, vaccines, everything is performed by biotechnology. You know that in the COVID, the vaccine recently it has come from shorter duration of time. They dispatched so many, the yield was so much. So from one gene they did too much of, from vaccine they gave lots of production. Okay, vaccines and antibodies. And in industries, your chemicals and everything I told you, we told you, right, glycerol which you wanted, like that. From industries they did so many chemicals, and also some of the enzymes whatever they required, they are all higher quantities, the production of more quantities now they are performing from biotechnology. So in the shorter period of time you are getting a more yield or more products. Okay, so let me tell you once again. Your blue is your marine, green is your agriculture, red biotechnology is nothing but your medicine. You will have vaccines here, and antibiotic, and your insulin also. And your white biotechnology is the industry, that is nothing but your chemical. So now let's move to your next slide. So this is your ancient biotechnology, how day by day the biotechnology developed. Ancient biotechnology, classical biotechnology, modern biotechnology, and in your agriculture we will see. So in ancient biotechnology, early age of food, you can term it as your bread we saw, beer we saw, wine we saw, and how the milk is getting into a curd formation, we saw that comes in an ancient. Later on, day by day, how did improvement biotechnology? Fermentation based off food production and medicine. Oh, they got it that if you add the yeast, it will get fermented. So like that, little improvement. And also in your medicines, I told you they founded penicillin using reactor. Next coming to your modern bio, that is your genetic engineering and molecular level manipulation. In genetic engineer, like your manipulation and engineering, what you are doing, you are taking a single gene, and from that gene you are developing so many products, okay, and so many offsprings, and for your performation use a PCR chain reaction or many products you are getting. Next one is in agriculture, crop rotation. So how it is helping you to, when they have put too much of crops which is affected by insects or all, you are controlling it by using a biotechnology products and by biotechnology methods also. And also in your food innovations, healthcare, and your animal husbandry, energy, and your environment management. So next in depth we will be seeing, okay. So what is that in agriculture it is taking place, medicine and all, okay. There are some different streams of biotechnology. See, there are nowadays, it's like biotechnology is like vast, okay. You have so many: genetic, protein, bioinformatics, immunology, plant biochemistry, cancer, environment, marine, nano, biophysics, pharmaco, cell biology, microbiology, medical and genetics. These are few streams of biotechnology. Every stream has a depth and a vast subject. Day by day they have been implementing, innovating everything. So got it? So now let's move on to your next slide. So I told you, right, next is your applications of modern biotechnology. Before going to the application, let me give you a quick recap on what we done, okay. So first, what did we do? Let me show you the slides. What did we do? First, we learned the term biotechnology, who was discovered, and what is biotechnology. We learned the definitions, and the next your your Paleolithic era started, and your lactobacillus in the curd, and beer making your with cereals and millets. Egyptian bread making was by your baker's yeast, and Louis Pasteur was the discovered fermentation and pasteurization. He was the father of microbiology, okay. And next is your bioreactor. You wanted to prepare a glycerol gas for your this thing, making of atom bombs, and modern father of biotechnology, and your scientist of genetic father of genetics, and this is your DNA, and your types of biotechnology: red, white, green and blue. And this is how your ancient, from ancient classical to modern, and application of crop improvement. These are few streams of biotechnology.